Handouts from last week as well in our discussion. The supplementary readings indicated on the screen for influenza. And then as a reminder, if you have questions about the grading, the deadline for reconsiderations is this Friday after class. Okay, are there any questions before we begin this morning? So if there are none, what I'd like to do is finish our discussion of the replication of influenza and then move to disease considerations. And where we left off on Friday is that in the case of influenza, RNA synthesis is within the nucleus of the infected cell. And there are three large proteins, keeping in mind that the genome of influenza is segmented, and we have these 8 segments of minus single-stranded RNA that are being transcribed by the virion-associated polymerase within the infected cell. And so what's truly novel is influenza uses cellular RNA as the primer to prime the synthesis of its own message. And what we didn't have on Friday is the slide on the left, which is figure 3 of your handout, that depicts how this happens mechanistically. So the 3 large subunits, the PA, PB1, and PB2, these 3 proteins associate with each other, and then they bind the virion RNA. So the genomic RNA, which is minus single-stranded RNA, is, as I indicated, sometimes referred to as vRNA for virion RNA. So those 3 subunits bind the virion RNA, but we don't yet have a primer. And so essentially the cellular RNA gets cannibalized to be a primer, and that's depicted at this step here. This would be the cellular mRNA, or on my schematic, it's this piece right here. And so the cellular mRNA is bound, and it's the PB2 subunit that binds the cap of the cellular RNA. And then the viral RNA, there's a lot going on here. The viral RNA, the polymerase subunit's bound at the 5' end, but then you have a looping where the 3' end interacts with the 5' end, where the polymerase subunits are bound. And when that occurs, then you do have the cleavage that's catalyzed by the PA subunit to cleave the cellular RNA. So you get just this little fragment of 10 to 15 nucleotides from the 5' end of the cell RNA that ends up being our primer that we can extend from to copy the transcript. So another way of looking at this schematically is for our virion RNA that's minus single-stranded, we have 8 different ones of this, keep in mind. And so the end sequences are conserved. So we'd have a U, C, G. This would be at the 3' end of the virion RNA, the minus RNA. So it would be at the 3' end. We have this conserved sequence, and then we have whatever the sequence is that makes it that particular gene. And then there's going to be a U track, and then some additional sequence in the 5' end. So what happens is that with the cellular RNA, we'll have a sequence of cellular RNA, that will include the 7-methyl G cap. What I'm writing in blue would be the cellular RNA. And then usually there's an A, and then some sequence X. And then you'll have 3 nucleotides that are complementary. So you'd have A, G, C. And then there'll be sequence on that makes this the cell mRNA. So this is the 5' end of our cell mRNA, and of course the 3' end, which is polyadenylated. And so for the viral sequence, the 3' has looped around at the 5' end to then position our 3' sequence, because we're going to transcribe 3' to 5', position it such that we have then the interaction with that short little region that's 10 to 15 nucleotides off the 5' end of a cell RNA. This is going to get cleaved by the endonuclease function of the viral polymerase. And so the PA protein catalyzes that cleavage. So this is cleaved by PA, and that's how we generate the primer that then is elongated by the viral catalytic subunit. PB1 is catalyzing that elongation. The cleavage was by PA, and then of course PB2 bound the cap to help set all this up. All right, so that's how we're transcribing within the nucleus. So the very end of the viral mRNA actually ends up being this cellular sequence. We're transcribing, and we hit a U sequence. And so when the polymerase hits that U sequence, it stutters. And as a consequence of that stuttering, as shown on your handout figure 3, so we've elongated, but when we hit the U sequence, the polymerase stutters and adds A's. So we'll stutter and add A's, which give us our poly A tail on the RNA. And so what does not happen in the transcription mode, or in the transcription mode, what does not happen is we don't make it all the way to the end of our minus genome template. As a result of the stuttering at the U, we polyadenylate, but we don't actually transcribe those terminal nucleotides in the transcription mode. All right, so we've generated these transcripts. They're then exported to the cytoplasm of the cell. And in the case of 2 of the genes, though, within the nucleus, as I gave you, and this is where we ended on Friday, of strategies of expression, 2 of the transcripts end up being spliced. And those are the ones that will encode the NS1 and NEP protein that we'll come back to. This is sometimes called NS2. And then M2 and M1 are also encoded by spliced transcripts, and the splicing occurred in the nucleus. The bulk of the full-length transcripts then are translated by using either 1 or 2 open reading frames. They give us our protein products, and among them is NP. So the NP protein that's made gets targeted back to the nucleus. So we've had then transcription, then we had translation, we've made our proteins, and the key protein that we need to make is the NP protein, because we then get back into RNA synthesis, but we're in the replication mode. And replication is going to allow us to make the full-length copy of the minus strand RNA. And that is a protein synthesis dependent event. We need NP in addition to the 3 polymerase subunits. And so when we are in the replication mode then, all right, you have this as figure 2 of your handout. When we're in the replication mode then, we'll make the complete copy of the minus strand RNA. And in order to do that, we need to have synthesized NP protein. That contrasts to in the transcription mode, which is the middle line here, which I sketched in a different way for you. In the transcription mode, we have the cellular cap, and then we have this conserved sequence where the priming occurred. We transcribed, but when we hit the U stretch, we polyadenylated. So the last nucleotides were never transcribed into mRNA. But in the replication mode, when we've made NP protein, this allows for the full copying of the minus template, so we end up with the complete strand, which of course we need in order to preserve the genetic information of the virus. So this event then has gone on. We've replicated and made then, as a product of the replication, first we make a full-length plus strand that then can be the template to make full-length minus strands, which are progeny genome segments. Okay, so this goes on in the influenza virus infected cell. We make then progeny genome segments. We've had protein synthesis, so we've targeted and trafficked back to the nucleus in addition to the NP that we needed for replication. PB1, PB2, and PA, our 3 polymerase subunits, are targeted back into the nucleus to generate progeny nucleocapsids, and this sets up a cycle conceptually similar to what we've had with our other viruses. And the bulk of the gene expression then is coming off progeny nucleocapsids, not the parental one that initially set up the infection. All right, so after a point then, when we've then generated the progeny nucleocapsids, we've got our 2 glycoprotein gene products, the hemagglutinin and the neuraminidase, that have been made, modified via the Golgi ER, similar to what we first saw in concept with VSVG. These proteins are targeted to the plasma membrane of the infected cell. So the next step that we're ready for then in our replication is assembly and release. And again, assembly is coincident with release because we generate the envelope of the virus particle by a budding process from the cell's plasma membrane. So influenza virus particles bud from the plasma membrane of our infected cell. And a key protein in this end stage, what we're going to have to do to assemble is these progeny nucleocapsids that were formed and functioning within the nucleus of the infected cell to generate our pool of mRNAs that are making the proteins. We're then going to have to target these nucleocapsids out of the nucleus of the infected cell to interact with the membrane where we've inserted the viral glycoproteins to then trigger this budding process and get our progeny viruses. And the protein NEP that in some texts is still referred to as NS2, is a key protein in this process. This is a nuclear export protein that targets the export machinery for the trafficking then and transport of the nucleocapsids from the nucleus to the regions of the plasma membrane that are modified by the glycoproteins, to viral glycoproteins that then end up being constituents of our envelope of the virus when it's budded from the cell. Okay, so what are the functions then of the glycoproteins? One of them we've touched on already, the hemagglutinin glycoprotein. And so the hemagglutinin, of course, was important in the initiation of infection. So glycoprotein functions. The HA protein is important in initiation because the HA protein is going to get cleaved to give us our two fragments, HA1 and HA2, as a result of cleavage by that cellular protease, and then these are retained with disulfide bonds. So the HA1 region will be important in our virus attachment, and that was to sialic acid residues. HA2 was important in fusion after we had acidification following receptor-mediated endocytosis, the fusion that occurred between the envelope and the endosome membranes, releasing that parental nucleocapsid. So the fusogenic activities in the region of HA2. The other function of the hemagglutinin is that it's the key neutralization antigen that we'll return to in a few minutes in consideration of vaccines. But antibodies that neutralize infectivity to a large extent target this region of HA, so it prevents the interaction with the sialic acid. Okay, how about the neuraminidase, Na? So the neuraminidase, as the name implies, is an enzyme. And this enzyme cleaves alpha-glycosidic carbohydrate linkages. And on your handout, on your handout you have the table that I gave you that lists the 8 genes and products. But you also have on your handout is figure 8 what the carbohydrate is. So we have a sialic acid linked to galactose, and that linkage can be either from the 2 to the 3 position, that's called alpha 2,3, or the 2 to the 6 position. It's that glycosidic linkage that's cleaved by the neuraminidase. And this turns out to be important in release of the virus. So the cleavage of that glycosidic linkage then has a role in enhancing release of the particles as they're budding from the cell. Because keep in mind, the sialic acid is where the HA bound to set up our infection, but we're producing particles that have to be released from the cell, and cleavage of that linkage by the neuraminidase is key in getting the efficient release of the progeny viral particles from the infected cell. Now it turns out that there are multiple neuraminidase enzymes in a cell, this neuraminidase, of course, is a viral gene product. It's encoded by one of our 8 segments. And as a consequence, it's been a target for drug development. And so there are 2 drugs. They're known as Tamiflu and Relenza. These are inhibitors of the neuraminidase. And so if you inhibit the neuraminidase, then you decrease the yield of virus from the infected cell. All right, so that's one drug target. The other drug target we've already discussed, and that's targeting the ion channel, the M2 ion channel, that's important in setting up early stages of replication because it's the allows for the fusion to release our parental nucleocapsid. All right, so those are roles of the glycoproteins and the 2 drug targets that have been developed and utilized so far. All right, now, how about influenza virus as an agent of disease, causing the disease flu? And why do flu epidemics happen? And why is influenza virus still more than exercise in molecular biology? It's a major threat for us. And so, in terms of seasonal flu, the estimates are still that there's in the order of 20 to 35,000 deaths per year from seasonal flu, just the seasonal flu cycles. And of course that brings substantial costs in many ways. In pandemics then, and what we mean by a pandemic is a global epidemic. And so in the context of pandemics, in the last century there were 3, and in 1918, as an example, the estimates were that there were 20 to 40 million deaths in 1918. So, huge. So, how do we get pandemics? Occasionally, there were 3 that we'll return to in the last century, and which pandemics are global epidemics. And so, in order to get an epidemic, you need some unusual ecologic situation present itself. And in the case of flu, there are kind of 2 ways that this happens. One is that you can introduce a situation where you introduce a large number of susceptible people, individuals, into an environment, a population, if you will, where you have a disease that's endemic. And so the susceptible individuals then will contract the disease typically. That's one way that can happen. And in 1918, that was a factor because there were large troop movements in the world, and so there were large groups of people moved to other populations where disease existed. For example, from North America to Europe. The other unusual ecologic situation can be if you introduce a new virus. And you have to introduce that new virus into a population of individuals that are susceptible because the population has no immunity because we have a new virus. And so that happened in 1918. A new virus arose. It also happened in 1957 and 1968. Or two other examples that I'll give you. All right, so for influenza virus, for human influenza virus, we have A, B, and C. So we have three types of our virus. We have influenza A viruses, B viruses, and C viruses. So influenza A viruses indeed do cause pandemics. You find influenza A viruses both in man and in animals. That's going to be key because the animals are the reservoir. B is exclusively man. And I'm not aware of any pandemics being caused yet by a B virus, but B viruses are increasingly problematic. And when we get to vaccines, you can see that the latest vaccine actually includes two different B viruses. And then C is very rare in terms of our viruses. So then, under the A-type viruses, we have these subtypes of our glycoproteins, the HAs. We have 16 different HAs, NAs. We have 9 different NAs. And so in man, in the human, in the context of widespread disease, the H types that have been effective are H1, H2, H3, and N1 and N2. For avian viruses, of which there's increased concern, H5 and H7, and also N9. Okay, so we have these different viruses. What then is the basis of getting a new virus? How do we get a new virus arising that then can lead to a disease situation? So the issue here is the strains are constantly changing. So why are the influenza virus strains constantly changing? In part, the reservoir is not limited to man, but it's animals, as you'll see with the reassortments that can occur. Okay, so this changing nature of the virus. There are a couple of ways that RNA viruses and influenza can change. And so you have on your handout, this I just told you, this is, we get in epidemics and pandemics moving people into an endemic region where you have disease or introduction of a new virus where you have no immunity. The key genes, among the key genes that are going to be important here, the hemagglutinin and neuraminidase glycoproteins. Okay, so you have this as figure 6 on your handout. So you get changes in our virus by a couple of mechanisms that I want you to know about. All viruses undergo mutation. And this is in part because RNA polymerase, when we're copying our RNA in the replication scheme that we've gone through, RNA polymerases make mistakes, and if a mistake is made, it's not corrected when you're replicating RNA. And the frequency estimates are that there's basically about 1 error per 10 to the 4th nucleotides of RNA that's made. So that's in one cycle. You make a mistake, but each time you copy, that mistake is amplified. So that's one source of error, of change. And this is believed to contribute to what we call antigenic drift. And what we mean by antigenic drift is the annual but independent changes in the immunogenicity of the hemagglutinin in the neuraminidase that's seen. And so what's shown to illustrate that point, the nucleoprotein is the reference, the antigenicity of that component. And so if you monitor the hemagglutinin and the neuraminidase, this is a function of time over activity, and there's a gradual change, annual gradual change. That's presumably because of the accumulation of mutations. They can alter the antigenicity, but they don't completely change it. So that's one contribution. The other is that you can have, with segmented viruses, as influenza virus is, because we have 8 segments, you can have genetic reassortment. That is, if you would have an infection with 2 viruses, you have reassortment of the genes, and you could get out a virus that has a novel constellation of those genes between 2 different strains. And if that happens, and we'll see some examples where it did, we call that change the antigenic shift. The major change, as illustrated in 1957, when there is a substantial change in the reactivity of both glycoproteins, and then in 1968, when only the hemagglutinin but not the neuraminidase changed. That's because of the reassortment of the genes, and you got a new gene that was different from the gene that otherwise was prevalent in the human population. And so last century then, examples of this, in 1918, this was the so-called Spanish flu. The virus was H1N1, and the origin is still a little bit controversial of that virus. But we ended up with 20 to 40 million deaths in 1918 from influenza. Then in 1957, when we had the shift of both the HA and NA, this was the so-called Asian flu. And we shifted to an H2N2 virus. The major virus that had been in the human population was H1N1. So you got a new virus that effectively circumvented the immunity that we had. And there were about 1 million deaths from that virus. And in this case, the reassortment involved the acquisition of 3 avian virus genes. The PB1, and then as the figure shows, both the HA and the NA, and those were derived from an avian virus. And then in 1968, it was the so-called Hong Kong flu. And in that situation, we ended up with an H3N2 virus. So in 1968, the hemagglutinin shifted, but the neuraminidase did not. But as I've already told you, the hemagglutinin is a major neutralization target when we have antibody produced. And so we had a new one that circumvented prior immunity. And in 1968, there were 2 avian virus genes that reassorted, PB1 and HA. All right, so then this has happened a few other times in 1977. Was back to H1N1 in the so-called Russian flu. 1997, the so-called Hong Kong flu again, and it was H5N1. And then in 2009, in California, Southern California, what arose was an H1N1, and in that situation, the reassortment was pretty novel, and you have that on your handout as figure. Yeah, so you have these figures on your handout, but the concept here now is man isn't the sole reservoir. We have nature, and as you can tell from my discussion with you, very often it's avian and it's waterfowl. And so you can have reassortments occur between these viruses to generate a new virus that would have some genes, and the key ones would be the hemagglutinin and neuraminidase from the animal, that virus circumvents immunity that we have, but yet retains other genes that you need for efficient replication of the virus, all right? And you have this figure 7 on your handout that I gave you the snapshot of, but over time, last century, this happened a few times, and the genes have been mapped, as I gave you, in terms of the origin of the reassortment, and it's in most instances avian.
So the reassortment is more likely to occur in the fowl than in the humans?
Not necessarily, right? Well, you said more likely. The key word is more likely. So the answer is yes, but these are spread. What's shown, the fowls are ducks, and these guys migrate, right? So you can have substantial change in spread of the virus, which is key. But for the 2009 virus, it's not fowl, it's swine, right? So it's not limited to avian. And so on your handout, which is handout 9. Yeah, so spring of 2009, a couple of very astute physicians in San Diego County and Imperial County, there were 2 cases that had no epidemiologic link. It was a 10-year-old boy and a 9-year-old girl that presented, and this was in April of 2009, and diagnosed as influenza, but different than had been seen. And within 4 weeks, and so this was in, as I said, San Diego County and Imperial County, within 4 weeks, that virus had spread globally to 41 countries. Really rapid transmission. And initially out of about 11,000 cases, there were 85 deaths. So rapid spread, but the initial indication then was not especially virulent. The genetic analysis was done. It was shown to be an H1N1 virus. That got people's attention because this was an H1N1 virus. when the analysis was done of the 2009 virus, it was complicated, and that's what's shown on figure 9 of your handout. Because it turned out to be a triple reassortment, which is kind of going back to his question. In this virus, the triple reassortment was swine, avian, and human. And 5 genes were swine, 2 from avian virus, and only 1 from the human virus. And so the 5 genes you have on your handout, but the swine were HA, NA, NP, M, and NS. The 2 avian genes were PB2 and PA, 2 of the polymerase subunits, and the human gene PB1. The good news is, in terms of the antivirals, the virus was sensitive to Tamiflu, the neuraminidase inhibitor, but the virus was resistant to flumidine, the M2 inhibitor. So in terms of vaccines then, so here, it was likely transmission from swine that was key. Right. Okay, so then in terms of barriers and what affects the transmissions of these viruses, because we have various genes that are changing, what are some of the barriers that are important in interactions and generation of the new strains? So one of the interactions is host-host. Man-bird. So here, migratory birds, poultry trade in Asia. You have to have contact between the hosts to get the spread. That's one factor. Or man, swine. For example, a pig farm, which is believed to be in Mexico where the initial transmission of the 2009 virus happened. So you have the host-host interactions that have to occur, and in some instances the virus is efficiently transmitted, and in other instances it's incredibly inefficient. And an example of that would be avian influenza A, the H5N1 virus. In 2006, there was an outbreak, 186 human cases, 105 fatal. So, highly virulent virus. And then in 2008, 391 human cases, 247 fatal. So this virus is highly pathogenic in its natural host. It kills birds, and it's highly virulent if it transmits to the human. But transmission is very inefficient. And you'll see in part why that is. But in the primary host, where the transmission is efficient, it's pretty virulent and it wipes out flocks. H7 N9 avian virus. In contrast to H5N1 that was highly pathogenic, this was lowly pathogenic in birds. So the birds spread with the virus without, in many instances, obvious signs of infection. And in eastern China, there were 80 cases and 17 deaths. So still visibly high, 20% mortality. Okay, so it can differ from virus to virus. And so then we come to host-virus interactions. What are the genes that are really playing a major role in will we have the infection and will we have disease? And so among the genes that have been identified, what viral genes are important as pathogenic factors? And so, of course, you'll name two of them from what we've already discussed today. One of the genes will be the hemagglutinin, because it's the neutralization antigen. But it also turns out that the receptor biology in terms of the sialic acid linkage is important. And so in avian species, so in birds, poultry, the linkage that's predominantly seen is alpha 2, 3. It turns out that the avian virus prefers alpha 2, 3, and so the avian virus prefers the linkage that's seen in the trachea and intestine of birds. So that's an important factor. Whereas in humans, the linkage that is predominant in the trachea and intestine is alpha 2, 6. And so the avian virus doesn't bind as efficiently with the alpha 2, 6 linkage. So aside from being the immunologic component, we have the receptor biology, if you will, that's key, and the glycosidic linkage affects that. And I showed you that on your handout someplace. Yeah, so at any rate, you have that with the schematic of the sialic acid on your handout. But in the human and in the bird, the linkage is different, and the HA prefers a different linkage. The other aspect of the HA is indeed it is a neutralization antigen. So HA is one key gene. Another key gene is the neuraminidase glycoprotein. And this has to do with virus release as opposed to setting up the infection for the HA. And again, you have the sialic acid glycosidic linkage factor coming into play because the avian virus neuraminidase prefers the alpha 2, 3 linkage as opposed to the alpha 2, 6 that we have as humans. The human virus prefers the alpha 2, 6. Another gene is PB1, the polymerase subunit that's evolved in the elongation reaction after priming with a cannibalized cellular cap. And for efficient replication of the virus in human cells, you need the PB1 gene from a human virus background. And that was depicted by the reassortments that we had that very often would have the human PB1. Then we need NS1, and I'll be finished in 2 minutes here. NS1 viral gene product we haven't discussed, but it affects innate immunity. The viral gene product NS1 inhibits the interferon response. And interferon is the cornerstone of innate immunity, and we'll discuss that with a whole lecture at the end of the quarter after we've seen all of our viruses. And then the last gene product is PB1F2, and I need 2 more minutes, and I think we should do that on Wednesday. Have a good day. We'll finish through on Wednesday.